No.
Oh my God.
All right, so
Fine.
yeah, we're going to finish up just a couple things. And I want to go back here. I know we already did this, but I want to just make sure something's clear because these terms are on your lab that we'll be doing, our enzyme lab. So we know that enzymes are made of proteins, and it's their shape that is important to how they function. We also mentioned this term of cofactor, sometimes called a coenzyme. Okay? The terminology on these is a little bit confusing because it's not really agreed upon in terms of cofactor versus coenzyme, but it's not really important for our purposes. Basically what you need to know about a cofactor or coenzyme is that it is an additional molecule that's required in order for the enzyme to work. Some enzymes don't have any coenzymes. They just work on their own as a single molecule and their shape is fine. But other enzymes may have one or more coenzymes or cofactors. And these are other molecules that need to bind with the original enzyme protein to allow it to function. Okay? And there's a couple words that we have for these things. Okay? So we have the cofactor, which is the smaller portion. Many of these are vitamins and minerals, and the reason that we need to have certain vitamins and minerals in our diet is because they act as cofactors for different enzymes that are important to how our body works. But the term is apoenzyme for the enzyme before it has the cofactor attached. And it doesn't work, or it may not work as well if it doesn't have that cofactor. Once the entire assembly is complete and you have the cofactor combined with the apoenzyme, the entire functional enzyme is called the holoenzyme. Okay, so that's where those terms, apo and holoenzyme. It's just, you're going to see it on the lab when we do the lab, so that's, I wanted to point that out. Something else I want to say? No, I guess not. All right. So we left off, we had talked about the different models and some examples of enzymes. And again, enzymes generally end in ASE. And we are currently collecting salivary amylase, and that's why you guys are spitting those cups. But an important thing to understand is how enzyme action is affected by various conditions. There's several factors that could influence how well an enzyme works. And the important ones that we usually talk about is our temperature and pH. Because how well an enzyme functions is dependent on those things. So generally, most chemical reactions, as the temperature increases, most chemical reactions will speed up. Because as we add heat, what happens to molecules? They move faster, and so often they're more likely to encounter each other. However, enzymes are made of proteins, and if we heat a protein up too hot, its shape changes. Okay? The protein can become what we call denatured, okay? And then it doesn't work anymore. It's basically permanently altered, and it can no longer facilitate those chemical reactions that it used to. Okay? So we have some terms. The optimum conditions for an enzyme is when that enzyme works the best. So the optimum temperature for an enzyme to work depends on the enzyme. But what would you guess the optimum temperature is for most human enzymes? Yeah, why? It's our body temperature. Those enzymes have evolved as our physiology has evolved. The enzyme action works best at our body temperature. So optimum is the most active. There's a point, okay, and if you look at this graph down here, this shows enzyme activity at various temperatures. And you can see that as we start adding and as temperature starts to increase, the enzyme works better, reaches its peak, which is its optimum activity. And if we want to know the optimum temperature for this enzyme, we just look at its peak, follow it down. It's about 40. But then as we keep getting warmer, this enzyme stops working as well until it completely stops working 100%, okay? That it's then inactive, okay? Then if the temperature continues, the enzyme can be destroyed and it no longer works. The same idea is true of pH. As acidity changes, the functioning of various enzymes changes. And there's some more variability here in terms of human physiology because some enzymes in our body work well at low pH, some work better at a relatively neutral pH, and some work better at a high pH. Okay? And so it just depends sort of where they're located. So you can imagine stomach enzymes, digestive enzymes in our stomach, they work best at what kind of acidity? A high acidity, a low pH, because those are the conditions inside of our stomach. Okay? Most enzymes sort of in our general body tissues work best around 7.3, 7.4, which is the general pH of our body fluids. But stomach enzymes work best at very low pHs because our stomach is hydrochloric acid in it, which brings its pH way down. Other enzymes, like in our small intestine, work better at a slightly basic pH, okay? Because that's the pH of the chyme as it passes through the small intestine. So you can see these graphs show a few different types of enzymes, and you can find each of them has a different optimum pH at which they work well. And last, the amount of enzyme or the amount of substrate are important. Okay? So as we add more of the substrate, okay, the enzymes facilitate that reaction and it goes up. But at some point, you can have excess of the substrate, and there's only so many enzyme molecules that can facilitate those reactions. If you have more substrate than you do those enzymes, well, they're sort of maxed out, and so you reach a sort of plateau when adding more of the substrate doesn't actually increase the rate of the reaction anymore. That's called the saturation point. So those various factors affect how those enzymes work. And this is, there's an enzyme in pineapple and some other fruits that is a protease enzyme. What does that mean? What do you think a protease enzyme works on? Proteins. It's a digestive enzyme for proteins, breaks them down. And so you cannot make Jello. You ever have those Jello molds that got chunks of fruit in it? Can't make it with fresh pineapple. Because fresh pineapple has those enzymes that actually breaks down the protein that allows the gelatin to sort of solidify. So you can make it with canned pineapple though. Why do you think you can make it with canned pineapple but not fresh pineapple? Yeah, but what about it? What about the process?
Heated.
Yeah, what the process of canning pineapple, what do you have to do before you can it? It's heated up to be pasteurized and then canned. So that heating process actually denatures the enzymes in the pineapple. And so the enzyme doesn't work anymore to break down proteins. When pineapple processing used to be done by hand, workers in those areas, their hands would become filled with sores because they're constantly handling these pineapples, and the enzymes actually started breaking down their skin, and so they had to use protection against that. Enzymes can also sometimes be inhibited by molecules that have a similar shape to the substrate. So we know because of the shape of an enzyme, fits together with certain substrate and causes a reaction. Well, if there's another molecule that has a similar shape, it can get into that enzyme and sort of interfere with its normal functioning. Okay? It blocks the active site from these substrates binding to it. And this is often one of the mechanisms that medications and drugs work on, is they are molecules often that are similar to other substrate or cofactors, and they interfere with the functioning of those enzymes. An example of this are what's called sulfa drugs. Sulfa drugs were the first types of antibiotics that were ever invented. You know when this was? When did antibiotics become common? in the 1930s, 40s. Sulfa drugs were the first. They were developed, I think, in the late 1920s and became commonly used in the 1930s. And sulfa drugs are basically mimic a compound that's found in bacteria. Okay, it's called PABA, and this is its structure. Okay. And it's used by bacteria to make folic acid. Folic acid is an important vitamin that we need. We get it in our diet, but bacteria, they have to synthesize it. They make it inside of their cells. But to do that, they need this molecule. Okay? The sulfa drugs, which is sulfanilamide, mimics it. It has a similar structure. It's a little different up here, but it mimics it closely enough that it binds with the enzymes that normally this molecule would work with. And that inhibits the action of that enzyme, making the bacteria unable to produce folic acid, which reduces their ability to reproduce. And so basically stops the spread of an infection, and eventually the infection dies off. So that's an example of a drug that works because of its similar shape to other molecules that interact with an enzyme. And many drugs work in a similar way. Any questions about enzymes? Okay.
That's it.
That's all.
That took 6 minutes.